Lovely day. And our last lecture together. Starting next week, Dr. Cumming will be here for 4 lectures, so he's here until April 2nd, which is the last lecture day. Potentially, that April 2nd will be a review lecture, depending on how many slides he gets through. So, while this is my last lecture, potentially I'm hoping to be back on April 2nd to do a bit of a review. Even if I'm not back on April 2nd, if Dr. Cumming needs that lecture to give lecture material, I will post review slides online, even if I don't have a review lecture on April 2nd. But hopefully, there will be a review lecture on April 2nd. All right? And then last Monday, the makeup exam for people who are qualified to write the makeup test, I should say. The makeup test is this upcoming Friday. Hopefully I've emailed everyone who is writing the makeup test, but in case you haven't heard from me, please contact me. If you've missed 2 or more tests and think you should be writing the makeup, contact me, please. All right, so we're talking about transport across membranes. Membranes are semi-permeable, but the cell needs to get large charged molecules across the membrane, things like glucose and ions and things. So there are mechanisms, protein channels, protein transporters that are in place to move material across the membrane. And we started talking about those, and we started off by talking about facilitated transport. So facilitated transport involves proteins in the plasma membrane, these are transmembrane proteins, such that whatever is being transported does not come in contact with the hydrophobic region of the plasma membrane. So the ion or whatever being transported is going to go through that protein channel or through that transporter and not come in contact with that hydrophobic region. So there are channels and transporters and pumps that we're going to talk about, and we started talking about different types of channels. So a channel is basically an opening that's in the plasma membrane that results because you have proteins, membrane proteins, that are allowing this open channel to exist. So there are channels that are going through the membrane, so material can move from the cytosolic to exoplasmic side of this membrane. Okay, facilitated transport does not require ATP. It's driven by concentration gradient. So the molecule has to be on higher concentration on one side, and then the free energy associated with higher concentration is what drives it to the outside. We use the example of the potassium resting channel. Potassium moving through this resting channel because it interacts with oxygens within that resting channel that are specific for its size. So channels, even though they're open structures, they are specific because of their spacing of oxygens and other molecules that allow for specific molecules to flow through. So the potassium resting channel is specific for potassium. Sodium can go through, for example. So potassium can go down its concentration gradient through a potassium resting channel. Okay, and then we sort of started talking about how that functionally can be relevant inside a cell. So we have this situation which I described last time, but I'll describe it a little bit better this time, where you have this experimental situation where you have this container, and you can think of it as sort of a cell, and then you put a membrane in this container. Okay, so this is a plasma membrane that's semi-permeable, but it's not going to be permeable to ions. Then you dissolve salts on both sides of this membrane. So you're dissolving sodium chloride and potassium chloride on both sides. But you're using different salts. So you can see on one side you have a little bit of sodium chloride, on the other side you have a lot of sodium chloride. On this side you have a lot of potassium chloride, on this side a little bit of potassium chloride. So you have these salts that have charged ions when you dissolve them in water, but the number of charged ions is the same on both sides. If you look at the molar ratios, there's the same number of positive charges, the same number of negative charges on both sides. Okay, so the salts are equal on both sides, the ions are equal on both sides. So when you look at the electrical potential across this membrane, there is no electrical potential because there's the same number of plus charges and minus charges on both sides, even though there's different salts. The charges across that membrane are the same. However, because you've dissolved different salts on both sides, on either side, one side has much more potassium. So on this side, it's 150 millimoles of potassium. On the other side, it's 15 millimoles of potassium. So there's a concentration gradient of potassium. So potassium wants to move down its concentration gradient, but if this is just a plasma membrane that's semi-permeable, it can't. It can't go through that membrane. It's charged. However, if you put a potassium resting channel in that membrane, if you allow potassium to flow through, the potassium will move down its concentration gradient. This is high on the left, low on the right. When potassium moves down its concentration gradient, it's going to take that positive charge with it. So potassium is going to move from the left to the right. It's taking that positive charge with it. So you end up with a positive charge being now present across this plasma membrane. And then you can measure that. You can look at the electric potential along that, across that membrane, and you can see that it's negative on one side and positive on the other. So you've created this concentration gradient of charge across this plasma membrane because that potassium moved across. Sodium can't move. There's high concentration of sodium on one side, but it can't move through that potassium resting channel. It stays where it is. So therefore, there's more positive on one side, more negative on the other side. Make sense? Right. So that is the potassium resting channel. Okay, so channels are open structures. Channels can be gated. We already talked about a voltage gated channel that can be opened and closed. So channels are sort of one way to do facilitated transport. They allow molecules to move down the concentration gradient. Another one are these structures called transporters. Transporters are sort of like channels, except they're never really open. Transporters kind of change the conformation to allow material to go from one side to the other side. So you're still using facilitated transport, you're still using a concentration gradient, you're not using ATP. And this is almost like a channel, except, as it says here, it's never really open because it's just a conformational change. So this example here is this transporter called GLUT1, and it transports glucose. So if you have a plasma membrane and you have a high concentration of glucose on one side of the membrane, it can get across that membrane using this transporter, because when it binds to one side of the transporter changes its shape and allows the glucose to pass through. So there's changes in affinity, but you don't have to memorize all that. Just the idea that this transporter allows glucose to bind on one side, it changes its shape, and glucose can exit on the other side of the membrane. So it's never really an open channel, but it changes its conformation because of glucose binding. So it is a ligand-gated transporter. Okay, it's only going to be changing its conformation when something binds. So you need a ligand, and it's a specific ligand. So GLUT1 is specific to glucose, and there's different transporters for amino acids, etc. Okay, so facilitated transport, so you have channels, gated channels, and transporters are all molecules moving down their concentration gradient. Right, they're specific to the molecules and they're saturable, so there's only a certain rate that they can go. Okay, so simple diffusion doesn't really do much in terms of the cell other than gas exchange. Facilitated transport, which we just talked about, involves these membrane proteins that allow molecules to move down their concentration gradients. Right? So we talked about channels that can be gated or not. So a gated channel would be the calcium release channel in the sarcoplasmic reticulum, and a non-gated channel would be the potassium resting channel we just talked about. Then there are these proteins called transporters. Transporters use facilitated transport, and they kind of change their shape to allow molecules to go through. So we just mentioned the GLUT1 transporter. It allows the molecule to go from the high concentration to low concentration. There are other transporters that allow two molecules to move at once. Okay? In the case of glucose, which is a single molecule, it's relatively easy to understand how that works. You're moving from high concentration to low concentration. There's a change in conformation. Okay? Because you're moving one molecule, these things are called uniporters. You can also have these transporters called symporters and antiporters. Here, more than one molecule is moving. One is moving down its concentration gradient, and the other one is moving against its concentration gradient. Okay? So again, you have a concentration gradient present. That concentration gradient is going to be put to work to move not only that one molecule, but another molecule as well. And they can be called symporters or antiporters depending on which way these molecules are moving. So if both molecules are moving in the same direction, it's called a symporter. Okay, so you have two molecules moving in the same direction, and we'll see an example in a little bit of that. Okay, but one of these molecules is moving down its concentration gradient. In this case, this red molecule is moving down its concentration gradient, and the energy that's associated with that is enough to move this other molecule against its concentration gradient. Okay, so two molecules are moving in the same direction, but one is going with its concentration gradient. It's the driving force, and it's allowing another molecule to go against its concentration gradient. And we'll see that in a little while when we talk about the sodium glucose symporter. You can also have antiporters. Okay, so antiporters are one molecule moving down its concentration gradient, which is easy to picture, but the energy of that is being used to move a molecule against its concentration gradient in the opposite direction. So antiporters molecules are moving in the opposite direction. So again, these are all movements that are driven by the concentration gradient. They do not require ATP. So none of these require ATP, they just require the concentration gradient. However, that concentration gradient needs to be established, and that concentration gradient is usually established through something called active transport, which we'll talk about now. Okay, so active transport involves using ATP hydrolysis, so you're using the energy of ATP to pump something against its concentration gradient. So these other types of transport here that we're talking about, symporters and antiporters, are often dependent on ATP-powered pumps doing something first. So usually you need active transport before you get to this co-transport. So these symporters and antiporters are called co-transport. So you need active transport, you need these pumps to establish a concentration gradient, but once that concentration gradient is established, then it can be used to do work, and you'll see some examples of that in a second. Okay, so now active transport. So active transport is you have membrane proteins that are involved in using ATP hydrolysis to pump molecules against their concentration gradient. Right? So there is a variety of different classes of these ATP-powered pumps. Okay, the classifications are somewhat confusing, but we're going to kind of keep it relatively simple, hopefully not tons to memorize, but a little bit. All right? So there are two classes of pumps that are specific to hydrogen. They pump hydrogen ions. Okay? So these are important in a lot of cellular processes, and I would like you to memorize at least where they're found. So there's something called the V-class pumps. That V-class pump that pumps hydrogen molecules into lysosomes. So lysosomes are acidified because there are V-class pumps on their membrane, lysosomal membrane, and they're pumping protons from the cytosolic side into the exoplasmic side. So the lumen of the lysosome is what's becoming acidified. That should make sense. So the V-class pumps on the lysosomal membrane are pumping these protons, these hydrogen ions, into the lumen of the lysosome. Third important pump that pumps hydrogen is F-class pumps. They are pumping these protons in mitochondria and chloroplasts. So you know mitochondria and chloroplasts have all kinds of gradients of protons across the membranes, and they're required for generating ATP. Well, F-class pumps are specific to mitochondria and chloroplasts. So I would like you to memorize both of these things are pumping these hydrogen molecules, ions, and the V-class pump is pumping, the V-class pump is found in lysosomes, and the F-class pump is found in mitochondria and chloroplasts. Okay, so that's what you need to memorize about those. We'll talk a little bit more now about these ABC type of pumps before we then talk about these P-class pumps. So let's talk about ABC types of pumps. So all of these transports require ATP hydrolysis, so they all utilize ATP in one way or another. But this class of sorry, did I go the wrong way? This class of pumps has a specific domain that's called an ATP binding cassette. Because they all contain an ATP binding cassette, they're called the ABC class of pumps. So ATP binding cassette. So this has a specific configuration of how they utilize ATP. Okay, there's many of these pumps, as it says up here. There's over 50 different types in mammals, and they can move a variety of things across a plasma membrane. So they can move sugars and amino acids and phospholipids and proteins and cholesterols and toxins. So there's many different types of pumps that move these molecules across the plasma membrane. Basically what they're doing is they're using ATP hydrolysis to change the conformation to grab something on one side of the plasma membrane and take it to the other side of the plasma membrane. So it could be anything from ions to cholesterol to a variety of other things. In terms of ions, one of these ABC class pumps is CFTR. CFTR is in the membrane of several tissues, and it's involved in transporting chloride ions. I'm not sure if Dr. Cumming talked about cystic fibrosis. Cystic fibrosis is a defect in this class of pump, this ABC pump that pumps chloride ions. The other thing that ABC pumps can do is some of them are flipases. So flipases allow these pumps to move things from one leaflet of the plasma membrane to the other leaflet of the plasma membrane. Remember I mentioned that the two leaflets in the plasma membrane, they have different phospholipids in them. There's different composition of the fatty acids that are found in the two leaflets, the exoplasm and the cytosol. And there's almost never any flipping between one side and the other. There's good lateral movement, things can move laterally within the plasma membrane, but they almost never flip within the plasma membrane. They can, and these ABC pumps called flipases can move phospholipids or sort of other fatty acids from one side, from one leaflet to the other leaflet. So ABC class pumps can flip things from one leaflet to the other leaflet. Okay, so the ABC pumps, pumping a variety of different types of molecules across the plasma membrane, including moving things from one leaflet to the other leaflet of the plasma membrane, or any membrane. So far so good? All right, before we talk about the last class of P-class pumps, which are pumping ions, when you look at cells, you see certain patterns in all cells. Okay, one is, that I described before, is that cells have very little calcium in them. So if you look at how much calcium is in a cell, whether that cell is a mammalian cell or a cell in an aquatic animal. So whether you're living in the sea, in seawater, or you're living on land, we have very little calcium in our cells. We pump calcium out of our cells for a number of reasons, as I mentioned, one of them being that phosphate is present, and you don't want the mineral component. The other thing you notice when you look at sort of ions in cell is that cells have high levels of potassium in them and low levels of potassium outside, whereas cells have low levels of sodium in them and high levels of sodium outside. So whether you're a mammal or an aquatic animal, you're pumping potassium into your cell and you're pumping sodium out of your cell. So cells have high levels of potassium in them and low levels of sodium in them. Okay, so cells are pumping calcium out of them, they're pumping potassium out of them, pumping sodium into them, into the cytosol. And this is all occurring through a class of pumps. So these are P-class pumps, and one of them is called calcium ATPase. So calcium ATPases are removing calcium from the cytoplasm. So the job of these things is to pump calcium out of the cytoplasm and sort of out of the cell or into the cytoplasm reticulum. So we've sort of mentioned a calcium ATPase, which is the muscle calcium ATPase that's found on the cytoplasmic reticulum. So during cell contraction, you have a voltage-gated channel that allows calcium out of the SR. Calcium moves out of the SR because of facilitated transport, so it's moving down its concentration gradient. It allows muscle contraction to occur. But as soon as the calcium is released, this calcium ATPase gets put into action, and it pumps the calcium back into the cytoplasmic reticulum. So it's pumping the calcium against its concentration gradient such that there's going to be almost no calcium in the cytosol and lots of calcium in the cytoplasmic reticulum. So you don't need to memorize any of these little mechanisms, but you need to memorize that two calcium are pumped out of the cytosol per ATP. In this case, for this pump, it's pumping it into the SR, but there's other ones that pump it out of the cell, out of the cell altogether. Okay, so the muscle calcium ATPase is pumping 2 calciums per ATP out of the cytosol into the cytoplasmic reticulum, and that's important during muscle contraction. Making sense? All cells also have a P-class pump called the sodium-potassium ATPase. So all cells are pumping 3 sodium out of their cytosol and 2 potassium into their cytosol per ATP. Okay, so in the end, because of the action of this pump, in the end, there's high levels of potassium inside the cell, high levels of sodium outside of the cell. The cell is constantly pumping sodium out, potassium in. 3 sodium, 2 potassium per ATP. So as it says here, all cells have low cytoplasmic sodium and high cytoplasmic potassium. Because of that situation, you can then put into place a potassium resting channel. So just like that experiment we just described, you have, where are we? You have high cytoplasmic potassium, you have lots of potassium inside the cell, you have lots of sodium outside of the cell. If you put a potassium resting channel in the plasma membrane, which we have, potassium is going to flow out of the cytoplasm. It's going to flow out of the cytosol. When it does that, it's going to take a positive charge with it. Okay, so the cell is constantly pumping potassium into the cytosol, just so it can then flow out of the cytosol and cause this charge difference. So because of the combined action of the sodium-potassium ATPase and the potassium resting channel, all cells have a negative membrane potential on the inside of the membrane and a positive membrane potential on the outside of the membrane. Right? So you're constantly pumping sodium out, potassium in, and that potassium that just came in is allowed to flow out because of that concentration gradient. There's a lot of potassium inside the cytosol, so it can flow out. As it flows out, there's a charge difference. All cells are doing this. Right? So that's where this secondary active transport comes in, this co-transporter. So now you have this charge difference across the plasma membrane, and it's going to allow you to do work with these symporters and antiporters. Okay? So this is called secondary active transport. So these antiporters and symporters, basically you're coupling 2 different molecules. 2 different molecules are going to move. This isn't just ions, but the examples we'll use are ions and sugar. So one molecule is going to move with its concentration gradient, and that concentration gradient has been set up already, and the other molecule is going to move against the concentration gradient. So this transport, this secondary active transport, does not require ATP, but ATP was required to form these gradients. And an example of that is something called the sodium glucose symporter. All right? You need to get material into the cell. Your cells select what's transported in them. Specifically your epithelial cells, which we'll talk about in the next few slides. You're eating food, you have tons of stuff in your stomach. Not everything gets into your bloodstream. The cell selects what goes into your bloodstream. It's going to select sugar, it's going to select amino acids, it's going to select fatty acid. All of that has to be transported from the lumen of your gut through the epithelial cell into your bloodstream and then wherever it goes. And it uses concentration gradients across the plasma membrane of that epithelial cell. So cells have high levels of sodium outside of them. Okay, sodium potassium ATPase, sodium is constantly being pumped outside. So there's a concentration gradient of sodium outside of the cell, but sodium wants to get in the cell again. Sodium wants to move down its concentration gradient into the cell. there is also a positive charge on the outside of the plasma membrane. The potassium resting channel leaks potassium out there, and because of that, there's a positive charge outside of the plasma membrane. So you have these two energies here. Okay, and I know there's a whole bunch of deltas up here, delta G's and stuff. You don't have to memorize any of the numbers, just appreciate that there's two energies here, these two sort of forces. You have sodium wanting to move down its concentration gradient, and you have sodium wanting to move down its charge gradient. Sodium is a positive molecule. There's positive charges on the outside, negative charges on the inside. So there's a certain free energy associated with sodium moving down its concentration gradient. There's a certain free energy associated with this charge gradient. And those free energies put together are quite a bit of energy. So there's a lot of energy associated with sodium wanting to get back into the cell. Its concentration gradient and that charge gradient. And that free energy is used to take another molecule with it. In this case, glucose. Right? So as sodium is moving down its concentration gradient, as you can see here, two sodiums are moving down its concentration gradient, it's moving glucose against its concentration gradient. This allows you to build up lots of glucose in your cell. So sodium is going to be moving in it, it's going to be moving into the cell down its concentration gradient because of these two free energies, and it's going to take glucose with it. And in the end, you can end up with high concentration of glucose within the cell. Okay, I'm going to show you a movie that will hopefully work. Variations of this movie are on Achieve. I should have used the Achieve stuff. The sound on this is awful, but let's bear with me and let's hope this works. Let's see if I can.
Energy is required for many key cellular functions. These cellular functions use the energy stored in the high energy bonds of ATP. ATP is generated using the energy from sunlight in photosynthesis, or by breaking down energy-rich foods in respiration. One important use for the energy of ATP is in the generation of concentration gradients across membranes. All cells have concentration gradients of ions across their plasma membranes.
All cells have concentration gradients.
This gradient consists of sodium and potassium ions. The gradient of sodium and potassium ions is generated by a membrane pump, the sodium-potassium ATPase. This ATPase pumps both ions against their concentration gradient, an uphill task requiring the energy provided by ATP hydrolysis. Specifically, the sodium-potassium ATPase pumps 3 sodium ions out of the cell for every 2 potassium ions that enter, coupling this with the hydrolysis of 1 molecule of ATP. The imbalance of potassium ions across the cell membrane can also create an imbalance of charge, called an electric potential. An electric potential is generated when potassium ions move outward, down their concentration gradient, leaving chloride ions behind. This creates an excess of positive charge on the outside of the cell relative to the inside. A concentration gradient of sodium ions and an electric potential power the transport of other molecules, like glucose, against their concentration gradients. The energy, delta G, for the movement of sodium is minus 3.06 kilocalories per mole. Via a 2-sodium glucose importer, this energy is coupled to glucose transport into the cell.
All right, hopefully that makes sense. You don't have to worry about the number of kilocalories and stuff like that. And obviously, when you look at the artwork, here's a importer, here's a importer, they've drawn it differently. It doesn't matter what it looks like. The important part is 2 sodium are moving down its concentration gradient, taking 1 glucose against its concentration gradient with it. And the energy is due to the fact that the sodium has not only a concentration gradient, but an electrical charge gradient that it's moving down. Okay, so now, 2 slides, the last 2 slides, putting a lot of this together. Okay, and that's using epithelial cells as an example, and this is your gut epithelial cell. So first of all, you got to understand sort of the picture and the sort of cellular structures here looking at this. So this is your gut epithelium, so inside your stomach and small intestine. This is the lumen, this is where your food is. This is the epithelial cell. Then over here is the bloodstream. Okay, you have to recognize that these cells have apical basal polarity. So that these little microvilli here, apical surface, they have a basal surface here, and then lateral side. So they have apical, basal, lateral. You should kind of be familiar with that. What is their job? Their job is to transport material across the epithelial cell. So you have food on one side, your bloodstream on the other. They want to select what goes through. They want to select specific sugars. They want to select specific amino acids. They want to select, other sort of useful ATP, whatever happens to be in your diet here. Okay? They do not want material to flow between the cells. They want to specifically select material. To do that, you're going to use a variety of transporters, including symporters. Right? So these symporters are going to be specific for different molecules. In this case, this is a glucose symporter, but you can imagine symporters for different amino acids. So to do that, what the epithelial cells do is they set up a system where all of them are doing the same thing. All of the epithelial cells are doing the same thing. All doing the same transport. So this involves the fact that there's a concentration of sodium and a concentration of charge across the epithelial cell membranes. So there's three things going on. One is there's a sodium potassium ATPase. All cells have sodium potassium ATPase. This is no different. Sodium potassium ATPase is pumping sodium out, potassium in. Okay, so inside the cell there's low sodium. So sodium wants to get into the cell. Potassium was pumped into the cell, but there's a potassium resting channel, so potassium flows back out of the cell. When potassium flows back out of the cell, now there's a charge difference. So the inside of the cell has low sodium and a negative charge. So now sodium wants to get back into the cell. Make sense? In your diet, you have lots of sodium. In your stomach, usually salt is essential. So sodium can come into the epithelial side of the into the apical side of the epithelial cell, and it can bring other molecules with it. In this example, it's glucose, but it could be an amino acid as well. So there are symporters sitting on the apical side of this epithelial cell membrane, and as sodium is coming into the cell, because there's low sodium inside the cell, it can take other molecules with it. In this example, this is a 2-sodium glucose symporter. Glucose is going to be transported into the cytoplasm of this cell. As you keep on doing this, you're going to increase the concentration of glucose in the cell to a point that there's going to be high concentrations of glucose in this cell cytoplasm. Okay, this example is glucose, but you can imagine it for any amino acid or sort of other molecule cells need. Because you have high concentrations of glucose in the cell, now you can use facilitated transport to get glucose out of the cell into your bloodstream. So you can use sort of transporters such as, this is GLUT2, but the same as the GLUT1, that facilitated transport where concentration gradient alone is good enough to get glucose across this basal surface of this membrane and out into your bloodstream. Right? So you set up a gradient first with active transport and this potassium resting channel, such that there's low sodium inside the cytoplasm and there's a negative charge. You then use co-transport for these symporters to get stuff from the lumen into the cytoplasm. Then once you have a high concentration of that molecule in the cytoplasm, you can use facilitated diffusion to get it across the basal membrane into the bloodstream. Aren't yourselves smart? Other stuff, though, that you have to kind of put together with all this. These cells are epithelial cells that are forming barriers. You do not want material to go between them. So you have tight junctions between them. Right? So material goes through the apical surface, not between the cells. You have microvilli. Those microvilli are these projections that increase surface area. They have actin in them. These epithelial cells want to be strong. They're probably connected together by desmosomes. Those desmosomes are likely hooked up to cytoskeletal components such as the intermediate filaments, right? So you can have cadherins present. You're going to have integrins present based on the basal side. Those integrins are going to be bound to extracellular matrix that's holding the basal surface together. You're going to have gap junctions. What are those gap junctions going to do? They're going to allow ions to move freely between these epithelial cells, such as all of these epithelial cells have the same charge. Not only are all the cells negative on the inside, but they all sort of have the same negative charge because of gap junctions. So you have this huge surface area that allows for transport because sodium can move through these, sodium can be transported along a large surface area because all the cells want sodium. That gap junction sort of facilitates that all of these epithelial cells sort of have the same low concentration of sodium and a negative charge. Okay, so there's a lot of things that have to go right here. There's a lot of cell adhesion going on, the cytoskeleton has to be going on. All of these membrane proteins have to be in the right orientation. Right, if they're going to be transporting things, they have to be in the right orientation. So if I tell you something is a PEG 2 or a PEG 3 protein, you should be able to figure out what faces what. Things are going to be linked to the cytoskeleton. So if you're disrupting different cytoskeletal components or extracellular matrix components, you should be trying to be able to figure out what is going on. So for the final exam, the questions for the final exam are going to be from these last few lectures, which are basically membrane proteins and the function of transport. But they're totally linked to everything else. Everything is linked to the cytoskeleton, cell adhesion, and extracellular matrix. So the questions can be about epithelial cells, but it can certainly ask you about gap junctions or cadherins or heavy desmosomes or something because they're important. Okay. Similarly with cell adhesion and things like cell migration, you need to kind of put things together that the molecules involved in cell migration are the same molecules we just talked about that are transmembrane molecules, including integrins. So while I might not be asking you specific questions about some of the proteins, like I might not ask you specifically about NDC80 and the function in kinetochore, certainly you need to appreciate that these cells are not undergoing mitosis and a bunch of other stuff. So you need to put things together. Okay, and as I said before, the best way to do that, and hopefully I'll be back for a review, is to put things in categories. So anything that actin is involved in, what does actin do, whatever it is. All right? I was going to show you the movie of the inner left of the cell again, and he's moving along, but people are restless and nobody wants to hear me. You can watch it yourself. Otherwise, I'll see you next a few Wednesdays from now, hopefully, on April 2nd. It is.
Yeah, right?
But like I said, it's cumulative, but most of the questions are going to be on the last stuff I talked about. It's just that you need to understand what actin is and what the desmosomes are and all that kind of stuff. So the questions might be on the new material. So most of the questions will be on the new material, but some of the answers are based on the old material.
So be interesting.
Yes, absolutely.
Thank you.
Sort of, but the charge would be on the other side.
Yeah, if it doesn't have the charge on the right now it's not.
You don't know. I wish I could tell you, man.